Hi everybody. This is the first of a series of videos that we're doing on histology. And this first video is just an introduction and overview about the microscope. So in this video, I'm going to explain to you what the parts of the microscope, and then in the next video, we'll talk about how to actually use it. So you may be tempted to turn this off and move on to the next one, but just remember that in the midterm exam, you will be responsible for the parts of the microscope, so you might as well listen. So the kind of microscopes that we use are called compound, whoops, let me get my pen working, compound light microscopes. And they're light microscopes because we use a light source to shine through and illuminate the specimen we're looking at. Okay, so that's a light microscope. It's a compound light microscope because it utilizes two sets of magnifying lenses. Okay, so that makes it compound. So our microscopes are binocular microscopes. So this area here is the eyepiece. Eyepiece. And the eyepiece has two little holes in it. So this, and this would be A here. So these two things are called the ocular lenses. And this is where your two little eyes are going to go look into the microscope there, okay? The ocular lens is one of those two sets of lenses. And the ocular lens is 10x in magnification, okay? Now, when you're looking at the microscope, I want to point out this part labeled G. This is the arm of the microscope. And down here at F is the base of the microscope. So when you get into the lab and you go get your microscope, you want to make sure one of your hands is on the arm and the other hand is under the base supporting it. The microscopes are pretty heavy and they're delicate, so you don't want to drop them on the floor. All right. The next part of the microscope, there's obviously an on and off button that you'll see, which was not shown here, but there'll be one of those. There is a little lamp here. It's just the light source, or the illuminator, and this is where the light comes from. And this thing at K is the light adjustment dial. The light adjustment dial. And that basically makes the light either brighter or dimmer. All right, the next thing we want to look at is this thing called the stage. The stage is where your slide is going to be sitting, and there is a clip on it, that secures it to the stage. So that's where your slide is going to sit. All right, over here at H, you can see H and I are this big knob and this small knob. This large knob is the coarse adjustment knob. And it moves the stage up and down. And it moves the stage up and down like far. You'll be able to move it all the way up until it stops and all the way down until it stops. The knob in the center of that, or right here, is the fine adjustment knob. And the fine adjustment knob does the same thing, but just a tiny, tiny bit. You won't be able to see the stage going up and down with the fine adjustment knob. Basically, you're going to use the coarse adjustment knob to get your specimen in view, and then you'll use the fine adjustment knob to make it real focused. Over here at K, and you can barely see it, this thing here, or excuse me, at J, is the mechanical stage adjustment knob. So we'll just call it the stage adjustment knob. And that knob is going to allow you to move the stage left and right and forwards and backwards. Because you want to be able to move the stage around so that the objective and the light source are on the specimen. If they're not on the specimen, you're not going to see anything. All right. The next thing we want to look at is this thing called the nose piece. So that's at B. And the nose piece is a thing that goes around. It can be turned. And on it are these things called the objective lenses. And this is the second of the two sets of lenses, are the objectives. There are usually 4, sometimes 3 objective lenses on the nose piece. And our microscopes have a 4x objective, and that's called the scanning lens. The scanning lens is basically looking at something from pretty far away. Remember that we have the ocular lens as well. And if you multiply the ocular lens magnification with the objective lens magnification, that's your total magnification that you're looking at. So if you're looking on the scanning lens, you're looking at something at 40x magnification. The next lens is the 10x lens, and that is low power. And when you move to the 10x lens, you're getting a little bit closer to the thing you're looking at. So your field of view is going to be more, it's going to be closer to the thing, and you're going to see more stuff. And it's going to be more, you're going to see the stuff better. All right, the next one is the 40x, and that is called high power. And when you move to 40x, that's going to get you even closer, so your field of view is going to be even less. But the stuff that's in the field of view, you'll be able to see more of and see it more focused. Okay. There is another objective, and that's the 100x, which is the oil immersion lens. And that objective is bringing whatever it is you're looking at really, really close. So close that you have to get the extraneous light out of the way. And that's what the oil does with the oil immersion. In anatomy and physiology lab for histology, you won't need the 100x objective. We can see everything that we need to see at 4x, 10x, and 40x. Okay? All right. Another couple of things that are on the microscope below the stage that aren't really pointed out in this picture. Under here, we have a little structure called the condenser. And the condenser basically directs the beam of light into your specimen. Okay? With that, and what helps control that, is the iris diaphragm. And the iris diaphragm controls the amount of light that passes through that specimen. Okay, so these two things together help for contrast. So if you're looking for specific things like the nucleus of a cell or something like that. You may need to utilize the iris diaphragm in order to provide enough contrast to see the things that you want to see. All right, so those are the parts of the microscope. Again, you will be responsible for naming some on the midterm exam. In the next video, we're going to talk about how to use the microscope. This is very important that you understand the steps involved in getting your slide onto the stage and getting it focused quickly, because you will have to do that during the exam for the midterm. So you want to be able to do that as quickly as possible. So I'm going to show you how to do that in the next video.